Hello, everybody. Welcome and welcome back to our STEM Lyceums. This is our session on college pathways, opportunities for research. So I'm Rebecca Carter, the program manager of the STEM Lyceums program, brought to you by the Center for Excellence in Education. I'm being joined today with Dr. Shannon Tyler, the program manager for the Teacher Enrichment Program. So, about the Center for Excellence in Education. It was founded in 1983 by Joanne DeGennaro and the late Admiral H.G. Rickover, who is the father of the nuclear navy and civilian uses of nuclear power, with a mission to nurture high schools and university scholars to careers of excellence and leadership in STEM. Our mission at CEE is to nurture high school and university scholars to careers of excellence and leaderships in science, technology, engineering, and mathematics, STEM, and to encourage international collaboration between and among leaders in the global community. Our programs at CEE include the Research Science Institute, or RSI, which is a highly competitive 6-week summer science and engineering program open to rising high school seniors. Juniors are also eligible to apply. RSI is the first cost-free summer science and engineering program that combines on-campus coursework in scientific theory with off-campus work in science and technology research. We also have USA Bioolympiad, or USABO, the premier U.S. biology education and testing program for high school students. Provides the motivation, curricular resources, and skills training to take them beyond their classroom experience to the level of international competitiveness. Since CEE began administering the USABO in 2003, every member of the USA Bioolympiad team has medaled at the IBO. We also have the Teacher Enrichment Program, or TEP. TEP provides opportunities for middle or high school teachers to connect with leading experts from industry and academia, explore cutting-edge research and development, and make meaningful professional links with direct benefits for teachers and students. And then we have STEM Lyceums, which is us right now. STEM Lyceums is a virtual club for high school students to meet once a month to learn about STEM career pathways from leading field experts. This also includes industry professionals sharing cutting-edge research and offer unique learning experiences, like scholarships and internship opportunities for high achievers. Please visit our website, www.cee.org, to learn more about our programs and opportunities. So I'd start with introducing our two speakers today for our session. First, we have Jasmin Modricker, Associate Scientist at Janssen & Johnson. Jasmin is a recent college graduate from Penn State University with a BS in Biomedical Engineering and a minor in Engineering Leadership Development. An integral aspect of her education was being part of a biomedical engineering research lab where she learned key laboratory skills studying regenerative engineering. Currently, she is an associate scientist at Janssen & Johnson, focusing on CAR T-cell therapy for cancer. We are also joined by Audrey Baker, a senior scientist at Janssen & Johnson, retired now, who has graduated from the City College of New York with a BS degree in biochemistry and also achieved a master's degree in biochemistry from Rutgers University. In addition to her education, Audrey gained certifications in biophysical techniques, such as light scattering circular dichroism, surface plasmon resonance spectroscopy, fluorescence spectroscopy, analytical ultracentrifugation, differential scanning calorimetry, and titration calorimetry. Audrey has contributed in the advancement of small molecule and protein therapeutics at Merck Sharp Dohme, GlaxoSmithKline, and Johnson & Johnson. She authored and co-authored several research publications pertaining to her research in pharma and developed and ran a program that introduced students to drug discovery processes. Currently, she is retired from Pharma, but actively engaged in promoting STEM education at various school events in Philadelphia, and also teaches Bible lessons to women and children. Thank you both for joining us. First, we'll be hearing from Jasmin.
Thank you, Rebecca, for that introduction. Thank you all for being here, and I really appreciate the opportunity to be here and talk to you about opportunities in research and also the research that I'm currently doing at Johnson & Johnson. So as Rebecca mentioned, and I just have my little blurb here about myself, I did recently graduate from Penn State University. Raise your hand if you've heard of it or you know of any relatives going there. It's a pretty big university. I really enjoyed my time there, and I have some alumni in the family who went there. I majored in biomedical engineering, and I did a minor in engineering leadership development just to get some leadership experience while I was in college. One big aspect is I joined a research lab there, as Rebecca mentioned, in musculoskeletal regenerative engineering, and we'll dive a little bit into what that means also. There's some big words. And then I'm currently in CAR T-cell therapy at J&J. We do a lot of discovery work, and I'll go over a little bit more about that and what that involved. I just wanted to give some background where I may be in your shoes back then. My passion for science and the STEM field started in high school when I was in AP Bio and AP Chemistry classes, which I really loved. I went to a small school, and a lot of the teachers mentioned that you could go be a doctor. Very general, very broad, and I didn't really know what that meant at the time. So I went to college and I went in a pre-med track, and I quickly changed it after I learned that there's many different career paths where you can be involved in science, you can be involved in medicine, in the STEM field, and even interact or be involved to help patients without being a doctor. So again, here's Penn State for anyone from the PA area or Philly area. So I just was involved in different clubs. Like I mentioned, undergraduate research was a big one. I was also involved in the Biomedical Engineering Society. If you're interested in the biomedical engineering major, that's definitely a good club to be associated with. And then I was just in some intramural sports teams like curling or cross country, which were fun. So here's some pictures about what research looked like at Penn State, specifically for chemical or biomedical engineering. So this was the labs that I was involved with. As you can see here, they're very pretty glass windows, more of like a molecular biology lab, I could say. We did some engineering work as well, but there's me in the right corner, actually, when I was graduating. That was the cell culture hood we had. So I would culture cells, like mesenchymal stem cells, and I got taught by postgraduate students, and it was just a really good experience where you could fail and it'd be okay, and they would help you through it. But these are just some images from a university perspective. I wanted to go over more in detail about how do you get involved with research at college. So those pictures I showed were really great and I really loved my experience, but how do you get there? So I definitely say it's never too early or too late to get involved. I reached out my sophomore year. I was actually in college during COVID year in 2020. So when we returned to campus, I got more involved in 2021. And I just went online and I just did some research around what labs exist, what am I interested in, and I reached out to a couple professors. So I just wanted to go into more about reaching out to at least 2 to 4 professors. I would highly recommend. It may be unlikely that you get your top one, and that happened to me. So it's just good to have a couple backups just in case, especially the ones that you're really interested in. I got a few recommendations from older students when I was looking my sophomore year, and they said to be sure to include specifics about the research that those professors are doing in your email to them. So you just send an email to these professors. For example, my professor, the lab that I was in, they did bio printing. And so I was specifically asking about that project, and he also had other projects as well. So what I would do would read through some of the research publications that they published to get a better idea. And this would be on their website, and you can read through it and get a better sense of what are the projects and what are you most geared towards. So I have a demographic of what I'm talking about. So right here, this is actually the website for the Penn State College of Engineering, the Biomedical Engineering College. And these are all the different research labs they have. And all of these sound amazing to me, and it was really hard to narrow it down. But I'll just go over the example for the lab that I was involved with. So I chose the Musculoskeletal Regenerative Engineering Lab. And as I mentioned, I did reach out to a couple other professors in different labs that did pique my interest. So once you click on that, and at least this website example, it'll take you to this website. So it just gives a generic overview of the lab and then who the PI, primary investigator, or the professor associated with this lab, who's Dr. Brown. When you click on this, you choose the research group website in this example, and then it'll take you to a lab website. So I assume most labs have their own lab website, and this is the page that you want to go to where they give you more of an overview of what's involved in the lab, who's involved in the lab. And here you can see there's a couple tabs: research, people, lab culture, publications, and there's a few others over there. But those are ones that I specifically looked at, seeing what is the research being done, who's involved in the lab, how many postgraduate students are there, or PhD students, what is the culture like, and also what are the publications. As I mentioned, I went through the publications to get a better sense of what the lab is like. So I was a part of this lab, and I'll go into a little bit of what it means and what we did. So this is the musculoskeletal regenerative engineering lab. The private investigator was Dr. Justin Brown. And just so we can break it down, the words. So musculoskeletal is the body's network of muscles, bones, joints, tendons, and ligaments that work together to provide structure, support, and movement. So you can see on the right a little demographic of what the muscles, bones, joints, tendons, and ligaments can look like from a skeleton perspective. And then regenerative engineering, which I really enjoyed being a part of, is the integration of material science and tissue engineering with STEM and developmental cell biology. And I know that's a lot to take in. And so on the left side at the bottom, you can kind of see this is what the lab was more about. So we focused on cell biology, which some of you may know a little bit about. So we looked at different cell types, specifically stem cells, like mesenchymal stem cells. We looked at signal transduction. What signals do they have? The signaling pathways. We looked at some biochemistry even. So we worked with different polymers. We looked at adhesion ligands. We also looked at soluble signals. And then we also looked more at physics and material science. As I mentioned, the polymer science that I was able to be part of. So we might have looked at porosity of these materials. We could look at the mechanics. Since we're looking at musculoskeletal and looking more at joints and muscles, we have to know the mechanics or mechanical engineering of how this will be integrated. So these are just a few things that we were interested in into pursuing in this lab. And at the bottom, we say we are focused on understanding the fundamental mechanisms by which biomaterial interfaces alter the proliferation, migration, and differentiation of mesenchymal stem cells to guide rational biomaterial design and facilitate the generation or regeneration of musculoskeletal tissues. So that also is a lot to read, but basically there's some tissues and some muscles that we can actually regenerate ourselves, which is really exciting for the human body. So that's something we're looking at, but we're using different biomaterials, specifically these polymers that interact well with our body, or can even be degraded into our body, and it's not harmful to us, that we can use in case this muscle tears or something like that. And then we use these mesenchymal stem cells to be put on these polymers, like these polymers that are healthy for the body, and then we see how those interact. So we have mesenchymal stem cells in our body, and so we see that interaction and see, is this actually good if we put this in someone's body? How does this interact? What are the mechanisms? What are the mechanics? What are the signals? Things like that. But I wasn't involved in all of that, but those are just some questions that we're asking in this lab. So here are some fun pictures that I actually took from my phone back when I was in school. And this is what we were really doing in the lab. And I know it may look, this TV looks really old, and it was really old. So we didn't have all that money in academics, but it was really, really exciting work. So on the left, as I was talking about, we were doing this 3D bioprinting. This is really heavy engineering work, and so we were using this with a polymer. So what makes bioprinting or 3D printing more bioprinting is that we looked at these polymers that we were using and injected that into the printing system instead of just using a different material. So we were specifically looking at this type of polymer that affected well with your body. So that was a lot of coding, just doing a lot of engineering work outside of your typical biology type of work. And so what we do is we create that polymer, and then we use that in the 3D bioprinting, and then we try to make these circles into a meniscus. So basically the project I was on was to create a one-layer meniscus using this polymer. So essentially, if your polymer is, or your meniscus is torn or it's partially torn, it's hard to heal. There is no surgery usually for if your meniscus is torn. So we were trying to look at what polymers will interact well with the body that potentially we could use instead to help heal it or to help just fix it. So here you can see is some curvatures of your meniscus that we were trying to create as this one layer base of your meniscus model. And then also when we'd create this, we would culture mesenchymal stem cells, as you saw me in the lab previously, and we'd look at these through the microscope. So this was a fancy microscope where we could look at different layers of the cells. So these are just mesenchymal stem cells you can see on the right. So these were just a few skills that I was able to gain being involved in this lab. As I mentioned, they do a lot in this lab, and this was just a few things, which I thought were really, really interesting. So then going into my career path, so I was involved at Penn State. This was my first year to third year in 2019 to 2022. And then after that, I actually got an internship at J&J in 2022, so my junior year summer, third year summer. And it was a 3-month internship in Springhouse, PA. I know I really, really enjoyed this. And I would definitely say that one big thing that how I got this internship was because of the research work that I did in the lab at Penn State. And so I had that on my resume. I definitely included all the laboratory skills because a lot of my peers in school while I was there, they didn't have all these skills because they might not have joined a research lab. They not have known what cell culture is or been able to handle that or any of these other skills that I was able to join. So that definitely helped me get this internship. And what the internship involved with was a lot of cell culture work. And that was really, it hinted at that I had that experience with these stem cells, and the internship was with stem cells. So not many people in their junior year have that type of experience. So it definitely helped me get here. Then after that, I just went back to Penn State and I graduated my 4th year in 2023 in May in biomedical engineering. And after that, my internship helped me to get a full-time job at J&J back at Springhouse in a different team, but in a very similar field of cell therapy. So like I said, the research that I did at Penn State really helped me get my internship experience. It's very competitive to get an internship at J&J, but I think taking the initiative to be part of a research lab is really, really critical to get you in there and really get you competitive with other people to be able to get that internship and actually land it. And then that internship helped me get my full-time role currently. So it's definitely a full circle type of thing. And then I'll go into a little bit of what industry looks like here at J&J. So we don't usually take many pictures inside the labs, but I found some images on Google. So J&J is involved with a lot of things, many, many different modalities, different targets we're looking at. So these are just a few things. We're looking at robotics. So I'm not personally in this lab, but they have robotic arms that move around and move different plates to different instruments, and it's really, really fascinating. Most labs have some type of robotic. Again, here's some cell culture hoods. So not much different from the academic cell culture hood, but we only had one in our research lab, and J&J has many. So it's nice to have more resources, but still a similar technique and skill that you can translate over. And again, here's just like a molecular bench. We use a lot of this at the research lab at Penn State to just do polymer work and like more chemistry work. But here at J&J, I might use it for more molecular biology. We might be running a PCR, digital PCR, running a gel, things like that. We might use the molecular bench for. And there's your microscope too, if you need to look at yourself. So now I'll go into what I'm working on at J&J and what the company is really looking at. So I'm involved in CAR T-cell therapy research. Raise your hand if you know what that is. I know it might be new to many of you and it's pretty new to the field, but it's really exciting work. So it's really looking at treating a different type of cancer. So it's a type of cancer immunotherapy where we take the patient's own T cells. So I'll go a little from the image shown on the left. So as you can see on the left, the top left, where it says remove blood from patient to get T cells. So we basically take a patient with cancer, we take their T cells, which is just a specific immunology T cell. In our immune system, we have these different cell types, and this is just one of the cells where our body uses a T cell to fight off a cancer or a virus or something that shouldn't be in our body. So we take out these T cells specifically, and then we just make these CAR T cells in lab. So basically, we're looking at this certain gene or this certain protein for this cancer, and so we're adding this CAR. And a CAR is a chimeric antigen receptor. So basically, that fancy word is that it's a synthetic protein where it can recognize and bind a certain protein target for this cancer. So we add this CAR, which can be reinserted into your body and target this certain cancer. So once we have that CAR within your own T cell, we grow a ton of them. We grow a lot and a lot of these T cells that are your own, like personalized to you, and then we infuse it back into this cancer patient. So essentially, it is your own T cell, your immune cell, that we take, we add this CAR to target this certain cancer that you're having, we grow them, put them back in you, and then they will bind to the cancer cells that you have because we make them specific for that cancer, and then they kill the tumor cells or the cancer cells that you have. So this is essentially training the patient's immune cell, an immune system, to target and destroy their cancer cells. So this CAR is trying to lead that effort to try and destroy this cancer within your body. And one of the potential benefits of this is to regenerate damaged tissue and reduce the risk of immune rejection. So usually when you add something to a person's body that they're not used to having in their body, your immune system rejects it. Similar to a virus or when you get sick, your body's fighting that. So we're trying to reduce it with a certain type of therapy, similar to like chemotherapy. This might be a different immunotherapy if you have a cancer. And you can see all the negative effects from chemotherapy that has on your body. It's pretty aggressive. So we're trying to reduce this and make it less severe on the attack that your body has on this. It can even treat a wide range of conditions by replacing this disease cells with healthy ones instead. And it offers a less invasive treatment compared to surgery or, like I said, chemotherapy. So how does this relate to J&J? So we actually have a current cell therapy commercial product available. It's called Carvykti. Not sure if you're aware of it, but it exists, and it's our one type of CAR T cell therapy that we have, and it actually fights multiple myeloma. So multiple myeloma, as you can see on the right, it usually targets bone marrow. So you can see you might have healthy bone marrow, and then you might have more diseased bone marrow. And it grows into these abnormal cells, and they have these abnormal antibodies where they can harm your bones, your kidneys, or usually your blood, where you'll see this cancer really present in. So Carvykti does that whole process that I just went over on the last slide, but usually with your blood or different areas that are affected from multiple myeloma. And then I'll show at the bottom here where it has this 5-step process of what it takes, kind of through the diagram that I showed, where we take those cells from your body, those specific T cells from that cancer patient, and that takes about 3 to 6 hours. You might be at a hospital or a certain type of cancer clinic where they're taking those cells out of you. And then step 2, where I said we're adding that CAR, we're genetically modifying your T cells. That might take up to 4 to 5 weeks or so because we're taking those specific cells from you and adding this CAR. So it does take a long time. We can't just make it a huge batch. It's just very personalized to you. Step 3, we have to pre-infuse you. So this is basically depleting your immune system. So we kind of have to infuse you for 3 days or so to make sure your immune system is not super healthy, because if it is, then if we inject this, your body might really fight against the therapy that's trying to help your cancer. So it sounds different, but we're basically trying to make your body less rejective of this new type of therapy. Step 4 is this one-time infusion. So after we create those CAR T cells and grow them a lot, then we infuse them back into you, and that might take up to an hour or so. And then step 5 is just monitoring you, seeing the effects, making sure everything's okay. And then that might be at 4 weeks, and even beyond, we'll just continue to monitor. And so I'll just go into a little bit about the current work that we're doing. So we help a lot with the CARVYKTI process, but we're also in the discovery space of cell therapy. So although we have this product that's on the market, we like to be invested in the discovery work as well, and how can we make it better? How can we make it cheaper for the patient? How can we make things more safe for the patient? Different questions that we're asking like that. So what I'm involved with is we're looking into different types of cancers, not just multiple myeloma, we might be looking at others. And also we're looking at different approaches for CAR T cell therapy. So another approach is using DNA editing or CRISPR editing. So raise your hand if you know what CRISPR editing is. I'm sure many of you have heard of it before, but that is really where we take this DNA editing. It's called CRISPR or Cas9 that you might be heard of, and it's kind of called this find and replace function. So you'll see we'll have a cell on the bottom, and the cell might be transfected with an enzyme containing a guide RNA and a healthy DNA repair template, as we might say, or a DNA copy, and then this DNA cutting enzyme. So basically what it does is we have this guide molecule or this guide RNA here, and we'll add that as specifically designed to find that target DNA strand that we might be looking for. So say if that's something exactly for the cancer that we're looking for, or another type of DNA sequence, this guide will directly go to that. And then we add this DNA cutting enzyme, and we'll cut that out, as you can see on the diagram. So we'll cut out that defective DNA strand or that certain DNA sequence that we don't want. And then we add in this healthy DNA strand or this DNA repair template, as we might call it. And that's exactly what we want. And then it's replaced with this healthy copy. So we might do this to target a specific cancer. We might do it to target a specific gene for a certain type of cancer, or different things like that. So that's really exciting work that we're looking into for CRISPR editing and different approaches for that to help improve CAR T cell therapy as a whole, and different areas that we can improve for gene editing. It can be used for different diseases as well. So that's just a little bit about me. And I really hope that people took away from this talk, and I really appreciate it. Thank you, and if there's any questions.
Thank you so much, Jasmin. It was very informative. We have a lot of questions rolling in through that. So we have some very excited students. Also very exciting that you mentioned CRISPR, as Dr. Zhang is an alum of CEE, so very much involved with CRISPR and
Awesome.
part of CEE's legacy. So very awesome to see that connection there.
Very cool.
All right, so now we have the pleasure of hearing from Miss Audrey Baker.
Good evening. Thank you very much, Jasmin. You can see in the blue box an image, and that's me. I was born in Jamaica, and this was taken around the time that I was around 13 years old. And at this time, I had no idea what I really wanted to do. However, few circumstances. The girl that's sitting to the left of me, to the fourth in this blue box, actually passed away about 3 years after this picture was taken. Her name was Winsome. She developed cancer of the knee. And, you know, Jasmin just spoke quite a bit about the wonderful advances that have been made in cancer research. Unfortunately, they were not available to Winsome. They chose to amputate her leg, and that gave her another 7 months of life, and then she succumbed to the cancer. And I think that kind of stuck with me in my journey, because I came to the United States and attended the University, City College of New York. And I started out as a chemical engineering major because many people knew that I loved chemistry and math and physics, and they thought, you know, you should really explore this field. But I really did not know what I really wanted to do. However, after a year in chemical engineering, I discovered a real deep love for chemistry, especially organic chemistry. So I switched my major to chemistry, still not quite knowing where my career would go with this chemistry. And there's something that I will suggest that you do when you get to college is keep up with the local news. And that's what I did. There was a campus newspaper, and in that newspaper was an article from Professor Donald Sloan. He was talking about his research in biochemistry. And as would have it, it was cancer research. Now, I had not heard much about biochemistry prior to this. So what I did is that I went to Dr. Sloan's lab and told him that I really enjoyed the research that he was engaged in, the little I learned from his article. And I made a request, a bit bold of me, but I thought I'd take a chance. I asked him if I could join his lab just to hang around to see what biochemistry was like. If he would allow me to look over his shoulders and the students who were in his lab as well, what I would do is wash the glassware, because at that time, beakers and pipettes, etc., had to be hand washed. But Dr. Sloan was so impressed, apparently, that he said he would actually give me a project in biochemistry. I was so blown away by that request. So, of course, I took him up on the offer, and he gave me a project working on an enzyme, orotate phosphoribosyl transferase. Quite a mouthful, especially for me, since I had not done any biochemistry. But life is about taking risks, and that's what I will encourage you to do as well. You see something and you feel some kind of passion for it. Go ahead and take the risk to find out what it is. So this was my first enzyme, and he taught me my first enzyme assay, where I was able to analyze the reaction of this particular enzyme. Now, this then led to me changing my major to biochemistry because I really loved it so much. And that's how I ended up graduating with a degree in biochemistry. And for you, I will say, make sure that when you go to college, if you find yourself in a situation similar to mine, you're curious about something, ask. Go ahead and ask. Don't be shy. Ask. And then the second thing is you have to show up. And that's what I did. When he said that he would give me a project, I made sure that I cut my lunch times and my free time short, and I showed up in the lab and I did the work. And then you have to deliver, because even though I did not know biochemistry, he expected high quality work from me. So that's what I did. I worked very hard in his lab and I delivered. So remember, please ask, show up, and deliver. And it changed my life. Now I'd like to talk a little bit about what research in industry looks like, a little bit of a piggyback on what Jasmin just told you. In the pharmaceutical company, our focus is on health care and the health of patients. And of course, the human body is very complicated and any part of it can break down. So what companies do is that they develop, test, market drugs that will alleviate the problems that we have. And the drugs that they develop must meet certain criteria of safety and efficacy as well. So very high standards. And that is the field that I worked in with my biochemistry. Pardon me, just a little frozen computer here. I'm going to give you a quick look at what the stages of drug discovery look like. I'm going to put it in 4 buckets, but they're probably around 12 buckets. So to discover a drug, you start out with drug discovery, then development, clinical testing, and finally, you hope to get approval to market this for use in patients. In drug discovery, the goal is to identify a target, a molecule that's associated with the condition or the disease, and then to develop a lead, which is the potential new drug. In development, you take the best lead and make it pure and safe so that it's hopefully efficacious. You then test it in laboratory animals. You generate quite a lot of data, which is scrutinized by the government. When they approve the work that you have done, they will allow you to conduct testing in humans. So that's the clinical testing stage. After all the work has been generated on the clinical level, and we're talking about years of work and billions of dollars, if the data generates what the scientists expect and indeed alleviate the conditions and is safe, it will then be approved for use in patients. So a little bit more about that. In drug discovery, what you would do is compare a healthy person with a person with a condition. And that's how you'd look for a target. For instance, the target might be a molecule in your blood or a chemical in your blood that is elevated in the case of the sick person, but is at a lower level in the case of the healthy person. That may be a good target. So target research will involve understanding how that target works in pathways in the body. If it looks like a good target for study, and if you generate a hypothesis that maybe restoring the normal levels of that target should alleviate the condition, then you will now develop a lead or a number of leads that will block or enhance or compete with that target. Again, the goal is to restore the levels or the concentration of that target in the body. The types of leads could be protein, could be a protein, or it could be DNA or RNA, or it could be a small molecule. Let's take an example of type 2 diabetes. In type 2 diabetes, the level of sugar in the blood is elevated for different reasons. There is type 1 diabetes, that is something that you inherit, but type 2 diabetes that develops with certain health conditions and over time. A target for type 2 diabetes could be insulin. For instance, could be that your insulin levels may be too low, or maybe the insulin that is in your body is not functioning correctly. So these would be potential targets. Another target that some of us may have heard of is GLP-1, very hot today. And a lead now would then block that target. For instance, insulin would be great because you would simply restore the levels of insulin in the body by administering insulin. So that is the oldest and most effective drug for diabetes. But there are other leads or drugs on the market, and you may have heard of Ozempic. How many of you have heard of it? Ozempic, Wegovy, those are big ones. They seem to advertise a lot. They are GLP-1 related drugs, and they related to diabetes as well. So I hope this illustrates what a target is and what the lead molecule could be. Here's another example. We just went through COVID, and we know that the COVID virus has spike proteins. And spike proteins are involved in entering the cells. So if you could block those spike proteins, you have a potential drug. You could use an antibody, but it takes a very long time to develop an antibody. Therefore, what we had from Pfizer and Moderna, we had RNA. RNA was made to the spike protein. When administered through vaccination, our bodies then allowed the expression of soluble spike protein, and then your or our immune systems would then generate antibodies to the spike protein, and that would then block the virus. So 2 examples of a target and leads. In my career in biophysics, I worked a lot on monoclonal antibodies, and this is an image of structure of a monoclonal antibody. So my degree was in biochemistry. However, I spent most of my career doing biophysics. And how did that happen? Well, I joined a biophysics lab. And what was helpful for me is that while I was in graduate school at Rutgers University, I actually did take a course in biophysics. So it was not a very painful transition to go from biochemistry to biophysics. I also discovered that I really loved biophysics, so that really helped with my transition. Just to give you a flavor of what biophysics involves, at least the techniques that I used, a lot of them are based on the properties of light. The sample showed here in a cuvette would interact with light. It could be visible light, it could be UV light, it could be even X-rays. When the light interacts or is allowed to interact with the sample, the properties of the light will change. And the changes in those properties give us a lot of information about the structure of the sample or the concentration of the sample or the way the sample, if it's a protein, let's say, interacts with another protein or DNA or RNA. So we get a lot of structural information from the interaction of light with samples. And the techniques that I used were circular dichroism, fluorescence spectroscopy, light scattering, and analytical ultracentrifugation, just to name a few. And all of them use light in some form or fashion. And that's an image of me. I am standing in front of an instrument. We call it a Biocor instrument. It also uses light. It's surface plasmon resonance. That is the technique. This is another technique. It's called calorimetry, where calories heat, and we're measuring the amount of heat that is released or absorbed when certain things happen. It's used to measure the stability of proteins, but it's also used to measure the interaction of proteins. For instance, if an antibody is to bind the spike protein in COVID, from a COVID virus, you could measure that interaction through calorimetry. Because when binding occurs, there is heat that is released or absorbed, depending on the mechanism, and that can be monitored. So this is just a flavor of what biophysics looks like. But my career continued because I next joined the regulatory department. In regulatory, I joined the Global Regulatory Affairs team for a short time, but it was wonderful because it gave me an understanding of how things are regulated. I'll take you back to the drug development process. And while we have molecules that we test in drug discovery, in development, the focus is really to take the lead, in other words, the best lead from discovery and take it through the process so that it can go in animals and humans. Before it goes from animals to humans, a lot of data is generated. All of the protocols, all of the results, everything, the information on the instruments that's used. Documentation is key. And this document, which is hundreds of pages, actually sometimes 2,000 pages in length, has to be scrutinized by the government. In America, it's the Food and Drug Administration. They go through everything and they critique everything. And when they're satisfied, only then is the drug able to be given to volunteers for clinical testing. After clinical testing is completed, and that takes years and billions of dollars, data is again generated, lots and lots of data that has to be scrutinized again by the government, the Food and Drug Administration, and they then will decide whether this data holds up to the promise that demonstrates that the drug is safe efficacious, and will work in the general population. This is where regulatory comes in, because the regulatory department of a company interacts with the government regulatory departments. They make sure that the company, Johnson & Johnson in my case, upholds its end of the bargain. In other words, we comply with the rules and the regulations given by the government to develop our drug. And on a global level, for instance, if we're marketing in Europe, well, we have another agency. We have the European Medical Association in Europe, and we have the Japanese and the Chinese. Every country has their own regulatory departments. So it's important to know what they require and to make sure that the company is complying. So the question comes down to you. You've heard quite a bit. And if you're interested in research, there are lots of areas within the drug discovery process where you need to do research. Lots of research occurs in the drug discovery area, but there's some research that goes on in drug development as well, although less so as you go forward because things are very much regulated before, in the clinical space. Lots of opportunities. It's a very rewarding part of science. I loved doing research, and you would love it too. So where am I now? I consider myself a STEM ambassador, where I hope to encourage students like yourselves to really consider jumping into the field. There's so much to do. As you can see, there are lots of areas that you can go in. There's a lot of innovation out there. And my question is, what might you discover? This is an image of Dr. James Andrew Harris, who back in 1969 discovered 2 elements of the periodic table. But the focus is now on you. So thank you so much for your attention.
Thank you so much, Audrey. I especially loved your point about ask, show up, and deliver. If there's anything that the students should take away and keep with them as they go into undergraduate, it's that, to ask, to show up, and to deliver. That was awesome. We're going to go ahead and move into our Q&A. I want to do a quick shout out to Kathleen, one of our students in our attendance right now. Kathleen, you are a superstar with all these questions. We are going to go through them all. All right, so going into our Q&A, first question I have up here, and this can go for either Audrey or for Jasmin, is there a good way for high school students to get involved into research before they go into undergraduate?
Yeah, I did a little bit of research before, and I think a couple of opportunities is definitely with different universities, especially even CE, I think has one too that was mentioned. But I think different colleges have opportunities for high school students, at least to be in an academic space where you can get more experience. So I definitely recommend that. That can really help, even if you're trying to get into, a really competitive college or university after high school. It definitely helps in that way to at least get some academic research experience.
I also agree with what Jasmin says, and I might just add that if you have a local science fair in your area, that is something that you should investigate. That might be a way to kind of cut your teeth in research. So do indeed check that. I know that in Philadelphia, where I am, there is a Philadelphia Carver Science Fair. And in Pennsylvania, there is a Delaware Valley Science Fair. So if you happen to be in that area, but if not, there might be others in your area.
Thank you so much for that. And let's see, to add in on that, another question is, can you share a bit about what you think is a good group culture and what to look for in a good principal investigator?
Yeah, I saw that question. It's a really, really good question. And that's something that I showed on the PowerPoint about going into the lab website and trying to get an understanding of each lab. One thing I've learned is definitely before you decide to, really go into that lab. You might talk with the PI beforehand, but before you're like, hey, I really want to join this one. Definitely talking to the PhD candidates and students there, even undergraduate students that might be a part of it. You might say, hey, there might be 3 PhD candidates. Can I just talk with them to get a better understanding? They should be really open and want to talk with you. If there might be like 3 or 4 PhD candidates, but only 1 undergraduate student, that might be a red flag to ask why are there not more students. Most labs have at least like an equal number to PhD candidates or even more undergraduate students. I know a lot of people want to be involved. So just getting an understanding from the students themselves in the lab and understanding their work schedule. I know some students, undergraduate students that were in lab very late hours with their PhD candidate, and that might not work with their schedule. So definitely understanding what is the expectation and what really fits with your schedule, and do you really get along with the PhD candidates, because they'll be teaching you a lot as well.
This also can go for both Jasmine and Audrey. So, why did you decide to go into industry rather than sticking in academia? And how does that feel different?
Audrey, do you want to go first on this one?
Certainly. At Rutgers, I got a flavor for what the environment in academia was like. And I think I preferred industry, even though at that time I really didn't have that experience. I think I preferred the focus on product, where you would focus on research in developing a project rather than just basic research, although there is a lot of collaboration, fair amount of collaboration between industry and academia. There definitely is. But the environment in pharma really worked out well for me. One of the things I really loved while working in pharma is working in teams. When you have a team, let's say diabetes, as I used illustrated in my presentation, if you're on such a team, that team is composed of so many different disciplines, so many different expertise coming together for one common cause, and you learn so much. It is just fantastic collaborating with each of the members. That has stretched me a lot, and my understanding of the therapeutic areas. I mean, I'm coming from a background of biophysics. So, for instance, the cell biologists are coming, from their expertise and bringing something else to the plate. So I really enjoyed just learning a lot from my team members.
Yeah, again, to piggy off of Audrey, I think one thing I also noticed in academia versus industry is the funding. Industry has a lot of funding, and like Audrey mentioned, we get to see the entire like product cycle, which is really, really exciting. And within academia, you kind of just dive into one specific project or one specific question you're asking. So it's very specific versus industry, you get to see a larger scope, and that really aligned more with what I was looking for rather than just diving into one thing. And even just to go back to the last question about the culture and what to look for, I just thought of it. Each PI and each lab has different types of funding. The lab that I was in regenerative engineering, we were able to have a partnership with a company called Medtronic. They look at like different biomaterials for musculoskeletal engineering. And so that was really helpful for our lab to get the funding, and we were able to purchase goods and purchase instruments and be able to go on about our project. But other labs might not have the best funding. So that's also just something to look for and even to take into consideration. I think it's good as high school students, everyone seems very interactive and really wants to, jump into research really early on, and that's amazing. I wish I was able to do that when I was a high school student, but that really gets you a feel of if you like research, do you like the academic space, especially as a high schooler, you can come into opportunities in academia. Is that what you want? Is that what you're looking for? That even will help you when you're going into college, looking for those labs. Like I said, just searching online, which labs are you interested? What are your specialties? And so you already have an idea of what you're looking for or the certain skills. So that really, really helps and sets you up if you want to be in academia, if you want to be in industry, and even specifically what are more of your interests too.
Awesome. And also building on top of that, what attracted you all in the professional realm to go with J&J as opposed to a different company?
That's a really good question. I think I always knew I wanted to be at J&J. I knew really early on into college. I think one thing that attracted me is that I always kept seeing news about them. I try to follow some like science news, STEM news, to see what's going on. And they were always involved in new diseases. They seemed to be very innovative. They wanted to take new approaches. They were diversified into like the engineering and maybe medical devices and also pharmaceuticals, and maybe a few other things. So it was really, really cool to see that a company was so diversified into different products, different diseases, cancers, things like that. So I really wanted to be part of a company where I feel like if I jumped into one thing, like I said, I'm in CAR T-cell therapy, and maybe I'm like, hey, in a couple of years, like maybe this isn't my thing. Maybe I want to go into medical devices and use more engineering, or I want to work in autoimmune diseases. You can really jump around because we have people and teams in all these different areas, which is really, really interesting, and not all companies have that.
And just to piggyback from off Jasmine's statements, I also respected Johnson Johnson, especially since I went to school at Rutgers and Johnson Johnson, well, their headquarters is actually in New Brunswick, New Jersey. So I was quite familiar with the company and a lot of the things that they were involved with. And what really got me there is that they had a strong commitment to developing biologics, and that's kind of an area that I really loved. I worked with small molecules, but my passion, my heart was still in biologics. So I was very happy that I got a position working in biologics at Johnson Johnson.
Awesome. And I'm gonna add on to that because I think this question kind of links up together. So we have, like we said, a lot of high school students that are interested in starting in research early. One student asked, does J&J take high school students if they have prior lab experience?
I know if you're interested, J&J has a careers at J&J website, and they'll have all their internships listed or different student opportunities. You can like specifically look for students. And within the requirements, it might ask if you're a college student, it might not. I definitely say don't let it hold you back. It's really good to apply, go through the process. Can even help you in a couple of years if you're a college student trying to apply and trying to interview. So I love the initiative. I would say go for it. I'm completely unsure if they have or have not before, but it doesn't hurt to try.
I agree. I think that you should at least go on the careers.jnj.com or something like that, what the website is, and at least set up a profile because it shows that you're interested. And even if you're a little bit younger than they had wanted for a certain position, your profile is there and they can see that you were serious from that time. So you'll also get a little bit of understanding of what sort of positions are there if you set up a profile and start scouting the site. So go ahead, it doesn't hurt.
That's an awesome piece of advice. Another student's asking, do you know of any high school internship or research opportunities around you? How would you suggest asking for an opportunity as a student? A really important skill to have, asking for that opportunity.
Yeah, I did a quick little search, and I know there's some opportunities at different universities nearby. I saw one at Drexel University, Temple University, Penn State. I'm sure there are many, many others. So I definitely recommend if you're interested or in the area to reach out to those universities. I assume it's just like an application. Usually if there's an email on there, it might say like a program coordinator, or something like that. I always love to just email them and just give a little information about myself. it's always good to just introduce yourself to the program coordinator as well as applying. And, sometimes I have a specific tie to a certain area. So be like, hey, I'm really interested in this research area, if that's one of the ones listed, or just in general, and just give like an overall understanding about yourself, saying, hey, I'd really love to go here, as a college student, or if you're interested in this field, just to give a better understanding of you and put yourself out there and maybe ahead of other students who might just apply and never email. It's always good to just do that little more effort.
I might add that it may not hurt to set up a LinkedIn profile. That is a professional site where people collaborate and, jobs are sometimes posted information on jobs. So you need to be very professional, but it is a great site for connecting with people in industry and also showcasing yourself.
I'll also add in a quick plug that with our March newsletter, I will be sending out programs that are summer opportunities and scholarships that are having March deadlines. So you should be receiving that newsletter soon if you have signed up with the STEM Lyceum's interest form. If you have not done so yet, then you should go to our program page and sign up on the STEM interest page so that you can get that mailer. Okay, adding on here, we have another question. Were there any particular extracurriculars that helped with your interest in research in high school?
Yeah. One thing I mentioned was the Biomedical Engineering Society, or BMES. I think most majors might have a little, I say, like science-y club with it. So that was just our society with the major. And I think one thing that really helped me, at least as I was able to connect with different PIs. We worked a lot in the BME lab space. So I was able to connect with different people within like the major up to first through fourth years. So it was really nice to connect with people that were older than me. They would tell me about their research. I was able to connect with different professors and meet with them if we had events. We might have had events with industry professionals, so I got to talk with them and do Q&A sessions really early on in my college career, as well as talk with different PIs and learn more about their research. So highly recommend, getting involved with different clubs and organizations that really pique your interest. If you have like a specific interest and there's a club for it, I would definitely recommend joining it, but don't join too many. But it's really good to join different ones that you're interested in. And even if you're able to get like a leadership experience out of it, I was able to become president of this club my senior year, and it really gave me that leadership experience. So when I was able to interview for my current role right now, I was able to say, hey, I have this research experience from Penn State. I also have my internship experience, and I even have some leadership experience from this club. And so they'll ask me questions in my interview, and I was able to answer because of the experiences I had as a leader in this club. So definitely recommend getting involved. It doesn't hurt you at all, and it can even help you with like teamwork skills, leadership skills, or any other those soft skills that you might not get outside of the lab.
And I did both of these as well, so it certainly works. I mean, I was a member of our science club at school, and we did a lot of science activities, but we also visited places like the blood bank. This is in Jamaica. And I also participated in our science fair as well. So these are things, any opportunity that you have to really get out there and network will definitely help you.
Most definitely. I think we have time for one more question before we go into our close. For our last question, are principal investigators willing to take in high school students, or I would say even undergraduate students, that might not have any experience in the research labs, but they've expressed interest in being a part of it?
Oh, yes. I had no experience going into being part of a research lab, and I would definitely say they want that. It's completely okay if you don't have any experience. It's amazing if you do, but I don't think it'll harm you if you don't. They're really looking just to train you. They know as an undergraduate student, or even a high school student, if you're applying for those like internships or those different programs as a high school student for a PI lab, they really just want you to learn. I think it just benefits you and it's also helping the lab out, but it doesn't harm you in any way. They will teach you everything. And like I said, one of the PhD candidates told me, and it stuck with me forever, is that's the time that it's okay to fail. You know, it's a little more harsh when you go into working in the industry, but that time is really just a form to learn skills. It's okay to mess up, and they do not expect you to have really much experience at all.
Indeed, you want to ask, let someone say no, but make sure that you ask and that you step up, you show up, and you deliver. Definitely. So I strongly agree that you really have to go out there and ask. You may not get paid, so that might be something to think about at first. Not all will be paid unless you're definitely fortunate. And the competition is stiff, so just be realistic, but don't let that stop you or discourage you. Do ask.
You for that wonderful piece of advice. I think they will take it to heart. And again, thank you to our sponsors, including the Claude Moore Foundation. They are the reason that we are continuing this cost-free programming to all of you students. So thank you to our sponsors. And once again, thank you to our speakers. Thank you to the audience for joining us. Please remember to complete the post-webinar survey. That is your attendance form for this event. If you are asynchronously watching this later, once it's posted on YouTube, I encourage you to complete this form. And remember to register if you have not done so. We have another webinar tomorrow, February 20th, with Dr. Lauren Myers. It is a can't miss opportunity. So thank you all. I hope you have a wonderful evening. And again, thank you to our speakers. It was awesome.